In chapter 3, we're going to stick with the innate immune system. We're just going to look at what comes about during the innate immune response, rather what types of things are induced and which cells are involved and which proteins are involved. So it's a little bit more in depth in content, but we'll move through it. This lecture is actually going to be divided into 4 different pieces because it is rather large. So there are a lot of slides in this deck, and so it'll take a little bit more time to go through. So if we go ahead and we just jump right in, we talked about in chapter 2 how a pathogen somehow has to get past that first barrier layer, whether it's the skin or the gut epithelium, but some way a pathogen breaches that epithelial layer and gets into the body's tissue or gains access to the internal tissues. When that happens, that's when that pathogen is going to be, or that foreign invader is going to be met with the army of the immune system, and that is going to be immediate. And so those immediate defenses of innate immunity kick in. So we looked at those. We saw that complement is going to be activated right at the surface through the alternative pathway. And there's going to be resident tissue cells that are going to be involved in helping to clear that pathogen. And with any luck, there's minor damage and that pathogen is cleared within the first couple hours of breach. But that isn't always the case. If the pathogen is not cleared from the body, then there's going to be another layer of innate immunity that is going to be induced. And it's considered still innate immunity because we're still in this non-specific recognition of the immune system. The immune system is not recognizing one pathogen over another, but rather it's just recognizing that something is in the body that isn't supposed to be there. And so this induced innate immune system actually will kick off after a few hours if that first pathogen if the pathogen isn't cleared during the first round. And this is going to include a lot more different cells. It's going to include a lot more different proteins. These proteins we call soluble effectors because they are soluble proteins that are sending messages and inducing cells to do certain things and make certain chemicals. And then hopefully, if all goes well, then with this induced phase of the innate immune system, the pathogen is eliminated and the infection ends. And then there might be some damage to the tissue, but hopefully it's not going to be too bad and it can be easily repaired, or at least rather quickly repaired. Inflammation is the key word of the innate immune system. We talked about that kind of, I think in chapter 1, we might have mentioned it, but now we're going to dive just a little bit deeper into what we mean by inflammation. Essentially, it's what the innate immune response does and what the innate immune response does really well. And so at first thing that happens here is in the first frame, we have some sort of infection. So somehow there is a break in this epithelial layer and pathogen are able to get in. So these little red guys are considered bacteria in this diagram. Okay, so when the pathogen gets into the skin, we call that an infection, or getting into the body tissue. That is called an infection. The first cells that they're going to be met with are going to be macrophages. Remember, macrophages are resident tissue cells. So they used to be circulating in the blood, likely as monocytes, that end up moving out of the bloodstream and then live in the tissues for this very reason, to quickly eliminate any pathogens that get through that first layer. So we have the infection occur, a macrophage is going to be there, and then it's going to internalize that pathogen and then produce what we call pro-inflammatory cytokines. Now, this is a new term. We've mentioned the term cytokine before. We talked about how it's a little tiny chemical messenger that is delivered between white blood cells. If we have cytokines that are pro-inflammatory, the definition kind of speaks for itself, meaning it's going to cause inflammation or at least promote inflammation to occur. So these triangles are a representative cytokines that are being produced, in this case, by the macrophage. Then what will happen, these little cytokines, these soluble proteins, are going to tell other cells what to do. So in this case, these little cytokines are telling the cells to come into the area, and they're also telling that endothelial lining to relax those tight junctions. And there's going to be a loss of integrity of that blood vessel. So this is the blood. It's kind of silly because it's white and the tissue is pink, and we're, it's kind of, but this is the bloodstream. And so blood will and fluids will rush out, cells will rush out of the vessel and then into the tissue. Well, when fluid rushes out of the tissue or out of the blood and into the tissue, there's going to be swelling. Along with the swelling, because of the extra fluids and cells entering the area, it's going to be hot because there's a lot of enzymatic action going on, and that's going to produce heat. This swelling is going to be painful because it's an extra pressure on the nerves in that area. And so there's four things that always go along, four indications, symptoms, signs that are inflammation, and it's heat, redness, pain, and swelling. Okay, so those are the key signs of inflammation. And in this gamish of inflammation, that is where the innate immune system magic happens, and that's what we'll talk about. So how do these cells recognize or know that something's a foreign invader rather than just maybe a microbiome, or maybe it's just a nothing, like some sort of a piece of wood gets in there without any bacteria? I don't know. How can it tell, right? And this is probably the cornerstone of the innate immune system's job, is to distinguish between non-self and self. And so we will talk about the difference between non-self and self for the remainder of the semester. What should happen in a healthy individual is that the immune system recognizes self cells and ignores them and says, oh, good, this is self, I don't have to worry about it. But it's able to recognize non-self cells or foreign invaders as being non-self and saying, you shouldn't be here, we're going to eliminate you. And so there's a lot of receptors that are involved in this recognition. More so in the adaptive immune system because it is a specific immune system, but in the innate immune system, there still are receptors that can at least non-specifically say self, non-self. Now, it's not going to determine anything beyond that. It's not going to determine, oh, you are a certain specific virus, but rather it's going to say you are not self. Those are NK cells, namely, if we're looking at the innate immune system. There are a few other cells that will be involved, but macrophages and NK cells have the bulk of these receptors that are utilized in the innate immune system. And what they're usually going to recognize are things that are going to be specific to pathogens or specific to non-self or non-human cells. And so it will look for carbohydrates, lipids, lipids, proteins, nucleic acids, sugar are found in both self cells and non-self. But those microbial cells, their carbohydrates, their lipids are just slightly different than what we see in human carbohydrates and lipids and proteins and such. And so based on receptors that recognize these macromolecules of microbes, macrophages, NK cells, and a few others are able to distinguish non-self from self. And then I should also add in altered self, because when we look at viral infections, it's going to be an altered self. So really, we're going to add in self, non-self, and altered self. You don't hear that as often, but it is an important distinguishing feature. So discrimination then between self and non-self and altered self will enable the innate immune response to be as precise as it can be. Now, because the innate immune system really lacks specificity, and it is kind of a like, hey, we have one game, this is the game we play all the time, and this is what we do. We have one defense, we run this defense no matter what type of offense we're seeing, right? But these cells are pretty good at being able to detect invading organisms, being able to detect cells that have been invaded by organisms or virally infected, but also tumor cells. And so this is also where we can see altered self, because a tumor cell is going to be a self cell, is just probably going to be altered in a way that looks different. And the immune system can pick that up. So here are some pictures that show these different situations. So we have bacteria that, so this little red guy is a bacteria cell. It has these proteins and carbohydrates on the surface that the macrophage has receptors for. Once those receptors are bound, then that pathogen is internalized, and then the macrophage is like, oh, I better send out cytokines. Okay, our NKs, oh, here's another macrophage too. We'll go down to this. Oh, it's just the second picture. Okay, so it brings it in. Yeah, tissue macrophage kills the bacteria and then will secrete cytokines. Now, in the case of an altered self cell, so we have, this is a self cell, but it has been infected by a virus. We will learn that virally infected cells actually put up little like flags that say, help, I'm infected, I need to be killed, right? And those are altered self proteins or altered self carbohydrates. And the NK cell or the natural killer cell will be able to recognize those altered self proteins and then actually add in death granules, essentially is what they are. And then that will kill the self cell. And then while the self cell is being killed, the virus is being removed as well. Okay. So within a population of cells, individual cells express different combinations of receptors, producing across the repertoire of macrophages and NK cells and other innate immune system cells to recognize potentially any type of pathogen or altered self cell that the body can encounter. So it's pretty great that the innate immune system, although non-specific, can still do a lot of recognition. Okay, so tissue macrophages. We're going to talk a little bit more about them in another lecture in this chapter as well, but they have a bunch of different types of receptors. So yes, they are going to be able to use these receptors to distinguish non-self from self, but there's a whole variety of different types of receptors. We're gonna look at a couple different groups of them in this lecture. We won't get too detailed on any of them because this area of research is rapidly expanding, and it is something that if you are in that area, you learn a lot about those types of receptors. But we're going to at least touch on them and introduce them to you in this class. So we have resident macrophages that are living in all the tissues, and they're ready and they're waiting. They're sentinel cells just waiting to see what's going to breach that epithelial layer and get into the tissue. And they have these receptors that would recognize these pathogens that get through, or these microbes that get through. And those receptors, when engaged by their ligand, will internalize that pathogen or that cell that ligand is on. And so this process is called receptor-mediated endocytosis. It's no different than the receptor-mediated endocytosis or phagocytosis that you learned maybe in a biology course in college or even in high school. I know that we cover phagocytosis in high school. We might not call it receptor-mediated endocytosis, but that's technically what it is. So what happens is you have this little pathogen that has these ligands all over the surface. And those ligands are going to engage with a receptor. So it's kind of a long receptor here on the macrophage. When that binding of that ligand by the receptor occurs, that's going to tell the macrophage to start to endocytose that pathogen or that bacteria cell. And so it will kind of fold in its cell membrane, creating then an endosome. That endosome will go into the cell, and then you'll have a fusion with this endosome with a lysosome. Sometimes this is called a phagosome. They're the same. And when that endosome or when that phagosome fuses then with the lysosome, they join together, then all of the acidic components and the other oxygen species, radical oxygen species, will combine and degrade whatever has been internalized. And then you have that phagolysosome when they join, and it breaks it down. Completely degraded. And so macrophages are amazing at phagocytosis, as well as our neutrophils. And we'll look at those in more depth in, later on in this chapter. So we have these different types of receptors. We have scavenger receptors, and we have LPS receptors. Lipopolysaccharide is what LPS stands for. Complement receptors we talked about in chapter 2. And then there's this group of toll-like receptors that we'll look at just a little bit more. But in all, these receptors are going to be very good at recognizing things that are different than self or different than human, actually. And so they're going to recognize patterns that are unique to microbes. And so we call them pattern recognition receptors, or PRRs. Now, if that pattern, is associated with a microbe, then we're going to extend that definition and we'll call it a pathogen-associated molecular pattern, or a PAMP. There's a lot of different types of PAMPs, like if you've taken microbiology, you've probably heard about peptidoglycan. You've probably heard about lipopolysaccharide. Those are unique to bacteria, and those are patterns that can be recognized by these receptors. Now, for altered self cells, they can put out signals that we call damage-associated molecular patterns, or DAMPs. And so when there's damage to a tissue, whether it's because of a pathogen infection or there's just some sort of damage, it could be a mechanical damage, maybe it's a tumor damage, those DAMPs can be recognized by receptors. And then the scavenger receptors are going to be very important in phagocytosis, because they're going to clean up dead debris, anything that is in the tissue that just needs to be removed and then recycled. So it can eliminate and clean up, but those are going to recognize patterns, is really what they are going to, what the ligands are. I've been using the term ligand maybe without defining it, but a ligand is what binds to a receptor. So you have receptor ligand binding. And so this table here shows the different types of receptors and then the different ligands that they will bind to. And so we have our LPS, we have our high percentage C and G DNA that is unique to bacteria cells. What else is in here? Peptidoglycan is in there. So things that are going to be associated with microbes more so than with self cells. So at the bottom here, we have our toll-like receptors, though, this bottom right here. Toll-like receptors, or TLRs, are probably some of the most characterized pattern recognition receptors. And there's a variety of different types of toll-like receptors. They're going to be named by number, and they're a family of signaling proteins that are going to be either embedded in the external lipid bilayer or membrane of the macrophage or NK cell or whatever type of cell we're looking at, or inside the vesicle, facing into the cytoplasm. And so toll-like receptors, depending on which protein we're looking at specifically, will recognize a lot of different components of bacteria and viral, as well as some fungal and parasitic patterns. So toll-like receptors are going to consist of two proteins that are dimers, and they can either be a heterodimer of two different TLRs, or they can be a homodimer of the same TLR protein. We'll look at some examples. And then they're going to have two different domains. They're going to have a signaling domain, which is the TIR domain. And then they're going to have a pathogen recognition domain, but like receptors, one that will recognize the pattern. So that's going to actually recognize the LPS or the DNA. And then we have this transmembrane portion that not only will serve to embed it into the membrane, but then to send signals down through into the cytoplasm of the cell so that signaling can occur. So here is a table then of these different, some of the major ones. There's more TLRs than just these, but you can see they're just named TLR 1 through 9. I think there's probably, or 10. Okay, there's probably 17 right now. I don't even know for sure how many TLRs there are. These are the most characterized. You can see that it shows what the receptor looks like, whether it's a heterodimer or a homodimer, and then if it's a heterodimer, what it's usually connected to. So TLR 1 and 2 are going to be a heterodimer. They're going to be back to back, and they are really important in recognizing bacteria and parasite patterns. Some of the bigger ones that we talk a lot about are TLR4. This is probably the one that's talked about the most. TLR4 recognizes lipopolysaccharide, or LPS, and that is going to be specific for any gram-negative bacteria. We find that it's embedded into the plasma membrane on macrophages, dendritic cells, mast cells, eosinophils, but macrophages and dendritic cells mostly. Some of the other ones that we talk a lot about is TLR9, another homodimer. This recognizes the cytosine guanine-rich DNA, which is commonly found in bacteria and some DNA viruses. And because these are going to recognize DNA viruses, which are going to be intracellular pathogens, they're located in the endosomes. TLR3 is another one that we talk a lot about that recognizes double-stranded RNA. Now, if you recall, double-stranded RNA is not a thing in human genetics. Double-stranded RNA is going to definitely signal that there is a viral infection. And so it's going to recognize RNA viruses, and because it's going to recognize intracellular proteins, they're going to be located in the endosome, facing out into the cytoplasm. So now, where this receptor location has everything to do with what type of pathogen they're going to recognize. And so we're going to go back to this concept that we talked about intracellular pathogens versus extracellular pathogens, and take a look at how TLRs engage, right? So TLR4, we looked at it on the previous slide. We saw that TLR4 was in the plasma membrane and it recognizes LPS. So if we take a look here, TLR4 is embedded into the plasma membrane, facing externally so that it can engage LPS from gram-negative bacteria, which are going to be extracellular pathogens. Let's go TLR3, for example. If we go back to TLR3, TLR3 is going to recognize double-stranded viral RNA. It's going to be located in the endosome. And so that's a DNA or an RNA virus. And so you can see that it is going to be in the endosome, which will then transmit, will pick up any viral DNA, will interact with the receptor, and will signal then down into the cytoplasm. So it can be located inside the cell in an endosome, or it can be located outside of the cell facing outward, depending on what type of pathogen. And then sensing of the specific ligand that that receptor is made for will then activate the cell to do stuff. And when we talk about doing stuff, especially in the innate immune system, what we're really talking about is turning on transcription of genes that code for cytokines. So depending on what type, so for the example that we have here, we have TLR4. TLR4 is going to recognize LPS, okay? It's going to turn on signaling, and then here's our signaling happening with a whole bunch of proteins that are involved in the signaling cascade. We're not going to worry about those in this course. We just have to know that there is signaling occurring. And what that does is it will turn on a transcription factor, which will then allow for the transcription and translation of cytokines. Then cytokines, as a functional protein, go out and send signals for stuff to happen. Okay, now you can see I kind of wrote over that it says pro-inflammatory cytokines. Let's take a look at what some of these cytokines are that can be produced. So again, they're small molecules, they're proteins, and they're going to be chemical messengers that tell cells either where to go, what to turn on, what they should be engaging in. And if we're looking at pro-inflammatory cytokines, we're looking at increasing of inflammation. And so, there's a lot of text on this page, but a cytokine then ends up becoming, it's excreted by a cell as a secreted protein, but then it becomes a ligand for a receptor that binds that specific cytokine. So for example, we have our macrophage up here. So this is our macrophage. Once it recognizes and internalizes bacteria and then breaks it down through the phagocytosis process, transcription is going to occur, and this may happen because of TLRs or whatever is going to be recognizing these specific bacteria cells. But then these different proteins are made, these different cytokines are made. Now the pro-inflammatory cytokines, and these are, you're going to want to remember, take some time to commit these numbers or at least associate them with pro-inflammatory cytokines. We're going to use them a lot, so it's probably going to happen naturally. But TNF-alpha, holy cow, if you're going to remember one cytokine that is going to increase inflammation, remember TNF-alpha. That is going to be very important in making those blood vessels permeable. And so they're going to enable cells and fluid and other effector proteins to be released into the area where the infection is occurring. And so these cytokines act in a very short distance. So this macrophage is going to be where the infection is happening. Those cytokines will be released where the infection is happening. And then the TNF-alpha will act in that area and release the swelling to happen in that area. Major one. IL-6 is important for the production of the heat that we see in inflammation because it actually induces fat muscles or fat cells to metabolize faster, and that increases temperature. CXCL8, not so much, I guess it's a pro-inflammatory cytokine, not because it increases inflammation in the sense of like more fluid or anything, but rather it recruits neutrophils. So CXCL8 is actually a chemokine that will tell neutrophils, hey, there's a smorgasbord of bacteria in this spot. You need to come here now and eat. And so more neutrophils will come into the area, which then causes swelling and pain and pressure. CCL2, not as talked about as much, recruiting monocytes, then they become macrophages into the tissue. And then IL-12 is another one it's another pro-inflammatory cytokine that activates natural killer cells to tell macrophages to become better. And it actually helps. So we'll see an interaction between an NK cell and a macrophage during a viral infection, where the macrophage will turn on the NK cell, and then the NK cell will then produce cytokines to further make the macrophage better at phagocytosis. So it's a communication. But TNF alpha, major pro-inflammatory cytokine to keep in mind. I know this is getting long. We have one more set of receptors I want to talk about, and these are NOD receptors. So NOD1 and NOD2 are family of another type of receptor, and they are going to be really important in recognizing bacteria and degrading them inside of the phagosome. And so they work alongside TLRs, just another type of receptor. NOD1, they're going to recognize the breakdown of peptidoglycan. So peptidoglycan is found in bacteria cells, cell walls. Gram-positive cells have a lot more peptidoglycan than gram-negative cells, but either way, if you have peptidoglycan, that's going to be a bacteria. And then, and NOD1 are going to be important for gram-negative. The equal sign means negative. And then NOD2 are also going to recognize peptidoglycan, but of most any bacteria. So just another type of receptor that can be used to help with the induction of the innate immune system. So TLRs, NODs. And in the end, what these guys are doing, though, is really getting the cells to be great at their work. So the pro-inflammatory cytokines will be turned on, with NODs also being engaged. And then let's see here, what do we got here? Nod-like receptors, yeah, okay, burst of IL-1 beta. IL-1 beta then is going to, IL stands for interleukin. It's still a cytokine, it's just interleukin. Cytokines kind of are used interchangeably. But it's the master regulator of inflammation. And so IL-1 beta then will act back on macrophages and then actually cause the death of a macrophage once it's done doing its work. Okay. Let's see here. This is the last slide I want to use for this lecture. We've been talking a lot about extracellular pathogens like bacteria, but when a virus infects a human cell, it's going to do so that it can use the host cell's machinery to make more of itself, right? That's what a virus does, is just survive. Well, when that happens, it's going to be producing nucleic acids for itself. Well, those nucleic acids are going to be kind of just floating around in the cytoplasm of the cell. And so there are intracellular receptor proteins that can detect viral nucleic acids and then try to interfere with this response. And so we have RIG and then RIG-like receptors. And they are going to hopefully interfere and block viruses from being able to fully replicate. So it interferes with viral infection. And then it can also tell neighboring cells like, hey, I have a virus, you need to gear up and really don't take anything in. If somebody comes over and starts knocking on your door, don't answer it because it's a virus. So it can act as an alert to other cells and then also tell cells in the immune system to come in, like NK cells, to go ahead and kill the virally infected cell. Oh, that was a lot. That was so thanks for hanging in with me on that one. But essentially, we have a set of receptors that recognize in a non-specific fashion self from non-self from altered self. And then once those receptors are bound and engaged, they're going to turn on the production of cytokines. Cytokines will then start cells to be doing their work, whether it's phagocytosis, whether it's interfering with viral replication, or whether it's directly killing an infected cell. kind of the nutshell. We'll pick back up in the second part of this lecture where we look at phagocytes.